Okay, guys, so welcome to another Introductory Anatomy and Physiology lecture. My name is Matthew Belzer, and most of you know me, but I don't know what semester this is going to be being shown, so I figured I'd introduce myself first. Now, as always, I'm going to walk you through what we're going through in today's lecture. So, we're not going to go into any degree of depth about the brain. The brain is a neurobiologically complex organ, and we could spend an entire 4 years talking about it and still not even scratch the surface. What we are going to talk about is the basic anatomy of the brain and the basic functionality of different structures of the brain. And that's going to be our focal point for today. So, what we're going to begin by is talking about how brain research kind of got a leg up to give you guys an idea of some of the types of questions I may ask you. We're then going to talk about the brainstem, which is a major region of the brain, the cerebellum, which is a major region of the brain in which there are specific structures you're going to have to know. We're going to shift our focus to the diencephalon, which is a major region of the brain that is composed of three primary structures I want you to know: the thalamus, the hypothalamus, and the epithalamus. We're going to cap off our discussion with the cerebrum. We're going to talk about functional areas of the brain, and then we're going to talk about subcortical gray matter, or gray matter within the cerebrum that isn't part of the cerebral cortex but serves some important functional roles. And that's what we're going to be doing today. So, as always, I've provided you with a set of learning objectives. You have this PowerPoint online available to you in both PowerPoint and PDF format, so you can print it out or you can have it up just on your computer so you can follow along. The other thing I want you to do is have the lecture handout either printed out or up on your computer so you can follow along and take notes as we go through them over brain anatomy and brain physiology, or the functionality of different regions of the brain. Now, in order to understand a lot of brain research and how we know so much about the brain, it's important to understand a pivotal moment in neuroscience. And that pivotal moment in neuroscience came in the mid-19th century, between 1800 and 1900. It was like September 13th, 1840 something that this happened, and I'm not going to bother looking it up. But there was a railroad worker named Phineas Gage. Now, to put brain research into context at this time, science knew very little about the brain, and we didn't have any effective methodologies really to probe the functional characteristics or to look at the functional characteristics of different parts of the brain. We just thought it was a grouping of matter, and we had no idea about it really. And then this railroad worker named Phineas Gage comes along. And Phineas Gage's contribution to brain science is kind of serendipitous for us, not necessarily for him if you read about his life. But he was a railroad worker, and he was filling a hole and temping it down with this thing called a temping iron. So what you're looking at here is a temping iron. And he was banging down on this, and there was gunpowder, and that gunpowder sparked, it ignited, and it drove that temping iron through his brain, right? It drove it right through his head. And what ended up happening is that temping iron went through regions of the frontal lobe. Now, the frontal lobe is responsible for an entire constellation of functions that we think of as being like executive function, personality. And Phineas Gage survived, but prior to the accident, Phineas Gage used to be a really nice guy, really easy to get along with. And after the accident, after regions of his frontal lobe had been destroyed, his personality totally changed. He went from being a really nice guy and easy to get along with to being really difficult to get along with, being really childish. His behavior became totally erratic. And that was of interest to science because scientists said, well, if that particular structure went through the frontal lobe of his brain and his personality changed, then there must be a link between the anatomical features of the frontal lobe and the regulation of the behavioral state of personality. And that led to a breakthrough in neuroscience because it gave scientists a methodology to start looking at different regions of the brain and trying to figure out what they do. In other words, if you want to know what a region of the brain does, one methodology you can use is to just destroy it and see what happens. So, for example, I'm going to talk about the brainstem, and I'm going to talk about nuclei in the brainstem regulating respiratory rhythms. Well, if you want to empirically demonstrate that that nuclei, that little chunk of gray matter in the brain, has something to do with regulating respiratory rhythms, you can go into something like a rat, and you can open up their skull, poke it out, and see whether the destruction of that particular gray matter area leads to functional disturbances with respect to respiration. If it does, what you've just demonstrated is a relationship between an anatomical region of the brain and a functional characteristic that that brain is responsible for regulating, in this case respiration. In other words, if you want to know what a part of the brain does, poke it out and see what happens, because it can give you insight. And those studies are called ablation studies, and they have led to a breakthrough in what we understand about the brain. It has established the foundation of a lot of our basic understanding about the way that different functional areas of the brain work. So Phineas Gage was really important. Now, when you think about the brain consists of 3 pounds of gray matter and white matter. There's billions of neurons, trillions of neuroglial cells connected in a network of such complexity that it boggles the mind. If you were to try and map out like a chunk of brain like the size of a grain of sand, and you were trying to figure out every synapse, every chemical, the electrical activity running through those neurons in that area, it would take up terabytes of data. Understanding the brain is a really, really difficult thing to do because it's such a complex organ. One of the things I want to highlight about the brain is we're going to be talking just briefly about this idea of connectivity. But connectivity is a way of stating that essentially every part of the brain is connected to every other part of the brain in some way, shape, or form. So there are relationships within the brain that don't seem intuitive, but you have to think that connectivity is the rule rather than the exception. So you may think, I thought this part of the brain did that. Well, oftentimes there are multiple regions in the brain that seem disconnected, but they are connected, and they can play, each of them play a different role in a certain set of functional tasks. So connectivity is essentially saying that your brain is highly intertwined, and all the different regions of your brain are connected to all the other regions of your brain. Now, when we're mapping out the brain from a basic functional perspective and an anatomical perspective, one of the first things that I'm going to ask you about are major regions of the brain. And the way we're going to define our major regions of the brain is we're going to go from caudal, meaning closer to the tail, to rostral, meaning closer to the head. That's another way of saying inferior to superior, caudal to rostral, kind of. And what we're going to do is we're going to map out the major regions first. So if I was to put a bracket around this whole region of the brain, the moment that the brain or the spinal cord passes through the foramen magnum of the occipital bone, it is no longer called spinal cord once it passes through the foramen magnum of the occipital bone. That's where the transition from spinal cord to brainstem comes, because the brain and the spinal cord are continuous structures. So the moment that the spinal cord passes through the foramen magnum of the occipital cord, we call it the brainstem. If I was to put a bracket around this entire area and say identify the major region of the brain indicated by the bracket, the major region of the brain indicated by the bracket would be the brainstem. Now, if the brainstem is made from 3 structures that we're going to be talking about, we're going to talk about where you find those structures and what the functional role of those structures are. The brainstem is composed of the medulla oblongata, the pons, and the midbrain. So those are the 3 structures that physically make up the brainstem, and we're going to elaborate on each one of those as we go over our discussion of the brain. The second major region of the brain I want you to be familiar with is this cauliflower-shaped brain region that you find on the posterior aspect of the brain that we refer to as the cerebellum. The cerebellum is this group of densely packed gray and white matter, really important for a lot of different functional things that we're going to talk about today. And that is a major region of the brain. Now, working our way rostral or up, when you look at this area right here, so I'm going to circle it, this whole area right here, this whole area right here is referred to as the diencephalon. And the diencephalon has 3 substructures embedded within it. We have the thalamus, which is this kind of circular grouping of nuclei located centrally in the diencephalon, forming the lateral wall of the third ventricle. So we have the thalamus. Now, inferior to the thalamus or below the thalamus, we have another really important brain structure called the hypothalamus. I want to note here that the pituitary gland can't be seen, right? So we're not looking at the pituitary gland right here. It's not readily observable at the moment. So we're not going to focus on that on this image. There are better images where I can show you where the pituitary gland is really clearly seen. And then back here, even though for some reason this particular image doesn't have it, this back here, this region back here is called the epithalamus. And what you're looking at right there, well, we don't talk about that in this class, so don't worry. So this region back here is called the epithalamus. And one of the key structures in the epithalamus is something called the pineal gland or the pineal body. The final major region of the brain that begins essentially that forms essentially the lateral walls of the walls of the lateral ventricle. This region of the brain that we're going to talk about that composes most of the volume of the brain, especially in primates like humans, is called the cerebrum. So we have the brainstem, the cerebellum, the diencephalon, and the cerebrum. Those are our major regions of the brain. If the answer is brainstem, cerebellum, diencephalon, or cerebrum, the question will be which of the following correctly identifies the major region of the brain indicated by the pointer. Now, those major regions are broken into different structures. And what we're going to do now is we're going to go over these structures from caudal to rostral, meaning from tail to head. And we're going to start our discussion with the medulla oblongata. So the medulla oblongata is really interesting. Now, I want to orient you to what you're looking at here. So you have the medulla oblongata right here. What we're looking at here is the brainstem. And this inferior portion of the brainstem that we're looking at right here, where you see these kind of bumps projecting out called pyramids, that's the medulla oblongata. Now, cranial nerves 9 through 12 originate on the medulla oblongata, and when you get into the peripheral nervous system assignment, you're going to understand how important cranial nerves are. What you need to know for an exam is the medulla oblongata contains a constellation of autonomic nuclei. So it contains autonomic nuclei, or little chunks of gray matter that are responsible for regulating specific functions, and it contains what are called vasomotor centers. Vasomotor centers control blood pressure. You then have cardiac centers, which play a major role in regulating heart rate. That's what we're talking about with autonomic. We're talking about those autonomic/visceral reactions or reflexes that we don't think about at a conscious level, but our body is constantly mediating and paying attention to. And finally, we have respiratory centers. Those are the three major centers in the medulla oblongata I want you to know. Vasomotor centers, which regulate blood pressure, cardiac centers, which regulate heart rate, and respiratory centers, which establish a respiratory rhythm. In other words, nuclei in the medulla oblongata essentially tell your diaphragm to contract and relax. In other words, it allows you to breathe. So it's a really important structure, right? So blood pressure, heart rate, and respiratory rhythms are established in the medulla oblongata. Nuclei for functional tasks like coughing, vomiting, swallowing are also located in here, so that's another thing to keep in mind. But think autonomic nuclei. Think of those functions that we carry out without even thinking about them, that our body is maintaining day in and day out. Another thing about the medulla oblongata, because it's essentially at the point that the spinal cord becomes brain, is all ascending and descending tracts have to pass through the medulla oblongata. Now recall a tract is a bundle of myelinated axons within the central nervous system, and many of these tracts, as they ascend up to the brain or descend down from the brain, actually cross over at the level of the medulla oblongata, and the crossing over of these tracts produces little bumps called medullary pyramids. Now, the artifact of crossing over results functionally in the fact that the right hemisphere of the brain is responsible for controlling the left side of the body, and the left hemisphere of the brain is responsible for controlling the right side of the body. There's adaptive significance to that, and we won't talk too much about that in this class from an evolutionary perspective, but there is. So we know the medulla oblongata contains autonomic nuclei, and we know that many of the ascending and descending tracts cross over in the medulla oblongata. The focal point is going to be the vasomotor centers that regulate heart rate or blood pressure, the cardiac centers that regulate heart rate, and the respiratory centers that essentially establish a respiratory rhythm, meaning they allow you to breathe. Now, intracranial pressure is the result of a buildup, essentially, of cerebrospinal fluid. And when there's too much cerebrospinal fluid for whatever reason in the brain, maybe it's a herniation of a brain region or whatever's happened, a brain injury or whatever's happening, intracranial pressure will increase. And when intracranial pressure increases, that puts pressure on the brain. When pressure gets put on the brain, it starts to panic and try and recalibrate and reestablish blood flow. And in that panic, there are three things mediated by the medulla oblongata that happen. So if anybody ever presents with an increase in blood pressure, bradycardia, which is a decrease in heart rate, and disruption of their respiratory rhythms, right? They're suffering from something called Cushing's triad, and Cushing's triad is really important to treat because left untreated, Cushing's triad can lead to death in a significant percentage of cases. So Cushing's triad is something they talk about a lot in, for example, paramedic medicine. And one of the things you do to treat Cushing's triad is you run an IV, like a hypertonic IV, and that can suck water out of the brain and essentially lower that intracranial pressure to alleviate pressure on the brain. So it's an example of how we apply our knowledge of things like tonicity to real world problems that you may encounter one day, for example, as a nurse. So Cushing's triad comes with a constellation of three symptoms, and it's really a disruption of what's going on in the medulla oblongata in many respects, especially that final step. And because the medulla oblongata is necessary for life, right, that's very important. That's an increase in blood pressure, a decrease in heart rate, paradoxically, meaning kind of weirdly, and a decrease in respiratory rate. And what you're trying to do in that case is suck water out of the brain to lower that intracranial pressure to allow the brain to recalibrate and reperfuse itself with blood. So it's a really, really dangerous condition. Now, what you're looking at here is an image taken from an ablation study, and this goes back to Phineas Gage. So let's say that we wanted to empirically demonstrate using data that heart rate was in fact regulated to a large degree by the medulla oblongata. So you've studied dissected portions of the brain and you see this little chunk of gray matter called the nuclei, and you say, I think that nuclei has something to do with maintaining heart rate. What you can do is you can take something like a rat and you can peel the calvarium or the skull cap back. You can take this little needle and you can poke that structure out and see, right, before was heart rate being regulated properly? Afterward, was heart rate being regulated properly? Oh, after destruction of that particular center in the brain, heart rate becomes dysregulated. And then you go, there's a link between that structure and that function. And that's one of the contributions of Phineas Gage. It was a methodology to start studying the brain that we still use today. So that's the medulla oblongata. Now, the pons, when you look at this bump on the anterior aspect of the brainstem and this region that interacts so closely with the cerebellum, what you are looking at is the pons. So if I ask you to identify the specific structure, that would be the medulla oblongata, and then you're definitely going to get function questions following that. If I ask you to identify the specific structure here, and we're looking at this bump on the anterior aspect of the brainstem, we're talking about the pons. Now, the pons contains a group of nuclei, or these little gray matter processing centers, called the pontine respiratory group. Whereas the medulla oblongata establishes respiratory rhythms, the pontine respiratory group fine-tunes respiratory rhythms. It essentially says, hey, stop inhaling, stop exhaling. And it uses data coming from higher order brain centers, coming from autonomic sensory centers. But the bottom line is the pontine respiratory group doesn't establish a respiratory rhythm. That happens in the medulla oblongata. It fine-tunes that respiratory rhythm, right? So it essentially says stop inhaling and stop exhaling. Another thing that the pons does is it mediates communication between the cerebellum and other structures within the brain, and that happens via what are called the middle cerebellar peduncles, which we're going to talk about. And cranial nerves 5 through 8 have a point of attachment on the pons. So 9 through 12, medulla oblongata, 5 through 8 on the pons. And when I say the pons is responsible for fine-tuning respiratory rhythms, let's take a look at a case study. So this individual comes into the doctor's office. Well, actually, no. Let me backtrack on this. This individual is brought to the ER unconscious. Consciousness doesn't spontaneously arise, so they put this person into intensive care. And within the intensive care unit, even though this person is breathing on their own, you notice that their breathing patterns are really erratic. So, first they're breathing really fast, and then they're breathing really slow, and then they're breathing really fast, and then they're breathing really slow. When you get that dysregulation of breathing, we call it kind of gasping breathing. Where you can bet your bottom dollar the damage is within the pons, because you're no longer able to fine tune that respiration. So you do the functional imaging on this guy and you see, oh, there's a tumor that's growing directly on the pons. You have this lesion or this damage to the pons. Now, that erratic breathing, that kind of gasping breathing, is referred to as Cheyenne-Stokes respiration. And I want you to see what it looks like, because I think it's interesting to apply some of the concepts we're talking about to clinical situations. So let's take a look at this video. Now, Cheyenne-Stokes is spelled down there for you. Now, if you look, you always determine respiratory rate by looking at the rise and the fall of somebody's chest. Now, if you look at this patient's respiratory rate who's in intensive care but not intubated, meaning doesn't have a tube allowing them to breathe, you'll see that he's hardly breathing at all. And out of nowhere, no stimuli, he starts breathing really, really fast. So the fact that he's breathing tells you that the medulla oblongata is relatively intact, right? And there's another thing in a second here that's going to tell you the medulla oblongata is intact. He's going to swallow, right? Now, notice that he went from breathing really fast, not in response to any kind of stimuli, now to breathing really slow again. And that kind of alternating hyperventilation, breathing really fast, and hypoventilation due to the inability to properly regulate respiration is referred to as Cheyenne-Stokes respiration. There are some not so sensitive colloquial terms or everyday expressions I've heard for this type of breathing. I've heard it referred to as guppy breathing. I've heard it referred to as the death rattle. So people who are presenting with that a lot of times don't have a good prognostic outcome, and that's dependent on the clinician and people with more information than me. I've just heard it referred to in different ways. Now, how do we know a lot of what we know about the respiratory regulation and the functional roles of the pons and the medulla oblongata? Well, there's a series of studies called the Lumsden cat studies. And what this particular researcher did is essentially he took cats, took the skull cap off, and removed the cerebrum. Now, the cerebrum isn't necessary to maintain many of our basic life functions. For example, respiratory rhythms and cardiovascular rhythms. Those are handled in deeper brain structures like the brainstem. And what he did with these cats is after he removed the cerebrum, he would evaluate their respiratory rhythms, and then he essentially took a deli slicer and went chur, 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 chur. And here you're looking at the pontine respiratory group. So before you get to the pontine respiratory group, right, breathing is totally normal. You cut through the pontine respiratory group, that's where you start seeing the Cheyenne-Stokes respirations, the erratic breathing patterns. You continue slicing through and you get through the medulla oblongata that has these really important respiratory centers that establish respiratory rhythms. The moment you get through the medulla oblongata, right, it's no longer gasping breathing, you're not breathing, and at that point you're dead, right? Or whatever you're talking about in this case is the cats essentially were terminated. But it gave us a lot of insight into understanding how the brain actually works. So does the ends justify the means? I don't know. That's a conversation for a different class. But it is a set of studies that played a pivotal role in our understanding of respiratory regulation. The next structure we're going to talk about is the midbrain. Now, within the midbrain, there are a few different specific structures that I want you to know. The first of those is called the superior colliculus. A lot of times I show the superior colliculus by peeling back the cerebellum and showing it from a posterior to anterior view. And this top bump right here, the superior colliculus, is a constellation of nuclei, meaning gray matter structures, that mediate what are called visual reflexes. So visual reflexes are things, can you track an object with your eyes? Can you see something coming at you and turn your head really quick? You don't think about it, but a lot of the movements of your eyes are just reflexive. They're being mediated by the brainstem. So visual reflexes are really important, and one of the things that you can evaluate with somebody who's inebriated is their visual reflexes. So if you've had a certain amount of alcohol, for example, that suppresses the activity of the superior colliculus and it makes it work a little bit slower. So if you ask somebody to track, right, your finger with their eyes without moving their head, they should have smooth movement of their eyes when they're tracking your finger. If their eyes are jumping from one point to the other, so they're like jumping to catch up, that's called saccades. And that is a telltale sign that something's going on neurologically or somebody is inebriated. So police officers are trained to look for that, and you cannot hide that. After a certain point of inebriation, you are going to present with that. Another thing to look at is the inferior colliculus. The inferior colliculus is responsible for auditory reflexes. If you jumped at that, what I did is I just activated a reflex center in your inferior colliculus. Auditory reflexes are things like hearing a bang and twitching your, contracting your muscles in such a way to take on a protective posture. Reflexes are just rapid, automatic, unlearned motor responses that are protective in nature. And that's what these reflexes are doing. So visual reflexes in the superior colliculus, auditory reflexes in the inferior colliculus. Think about that walking up behind somebody with a gun. If you did that and you pulled the trigger and that person didn't have a reflex like that, that might indicate something about the functionality of the inferior colliculus. Finally, we have a region in here that you can see a little bit better from a cross section, and you don't have to know it anatomically, but just functionally I want you to be familiar with it. It's called the substantia nigra. And the substantia nigra, right, contains these neurons that produce dopamine. Dopamine is really important in movement. It's a chemical signaling molecule called the neurotransmitter, and it's really, really important in regulating movement. And in diseases such as Parkinson's disease, neurons in the substantia nigra that produce dopamine that works in different regions of the brain can become damaged or destroyed, and then you get this dysregulation of movement that produces a tremor that's common or characteristic of diseases like Parkinson's disease. So the substantia nigra is intimately involved in the fine-tuning of movement. It also communicates with the cerebellum via what's called the superior cerebellar peduncle, which is a white matter pathway leading out of the cerebellum. Not a huge focus on the test, but something I want you to be familiar with. Now, cranial nerves are presented within the peripheral nervous system lecture. I highly recommend learning a mnemonic. Mine is on Old Olympus's towering top, a friendly Viking grew vines and hops. With the exception of cranial nerve 1 and cranial nerve 2, all of those cranial nerves have a point of origin on the brainstem, and many of the nuclei in the brainstem are just called the nuclei of the blank cranial nerve, and they're mediating the functional characteristics of that particular cranial nerve. So when you think about, for example, the vagus nerve has a point of origin within the medulla oblongata. And when you look at the sensory inputs and the motor outputs of the vagus nerve, when you think about the medulla oblongata controlling blood pressure by having vasomotor centers or heart rate or respiratory rate, you can see that innervation. There's a structural innervation or control of those structures that allows the medulla to do that. That's the peripheral nervous system, in this case the 10th cranial nerve, mediating communication between the central nervous system and the structures that it controls. So remember that the peripheral nervous system transmits sensory input to and motor input away from the central nervous system. So know those cranial nerves. You're going to have a detailed lecture about them. Now, the next functional or major region of the brain that you need to know is called the cerebellum. And the cerebellum consists of some of the most densely packed gray matter in the brain. It's an extraordinarily important brain structure. Some of the functional roles of the cerebellum is it fine-tunes skeletal muscle contraction. So the cerebellum doesn't initiate skeletal muscle contraction. You can initiate a gross skeletal muscle contraction or just a really rough contraction independently of the cerebellum. But what the cerebellum does is it fine-tunes skeletal muscle contraction by dampening down the activity of accessory neurons, right? So it can provide corrective feedback to regions of the brain like the primary motor cortex. But the bottom line is it fine-tunes skeletal muscle contraction. So if you're trying to thread a needle, for example, your cerebellum is going to be kicked up and in full throttle because you need a lot of fine-tuning of muscle contraction to do that. Muscle memory is largely, it's also called procedural memory, is largely a consequence of the functional characteristics of the cerebellum. So things like walking, you forget that all of us had to learn how to walk, but once you learn that kind of procedural memory of putting one foot in front of the other, right, a lot of that, those motor tasks are predominantly stored within the cerebellum, things like playing a musical instrument, riding a bike, riding a snowboard. What we consider muscle memory, that's largely a consequence of cerebellar activity. Proprioception. Proprioception is knowing where your body's position is in space. So you have proprioceptors located all throughout your body, so you don't need to have your eyes open to know that your arm is above your head at any given point in time. And a lot of those proprioceptive pathways are being interpreted or processed within the cerebellum. It's really important for proprioception. So one test we do for proprioception is we'll ask somebody to close their eyes, and then we'll lift their finger up and say up or down. If you say up, down, up, without seeing it, you know the position of your finger because you have these proprioceptors giving you information about your body's position in space. Finally, balance and equilibrium. So being able, for example, to walk a straight line without woozing all over the place or walking heel to toe. And one of the things about the cerebellum is a lot of us have heard of what's called the field sobriety test. That's where a doctor, one of the quintessential tests of cerebellar function is the finger to nose test. So taking your finger and putting it to your nose, right? And can you do that with your eyes open? Can you do it with your eyes closed? Do you have trouble doing that? That's fine-tuning skeletal muscle contraction, right? Putting your finger to your nose involves a direct and very specific trajectory of your finger, and you need to fine-tune your muscle contraction to do that. Alcohol, one of the regions alcohol hits the hardest is the cerebellum. So if you're unable to do that because alcohol has shut that region of the brain down, it may indicate that you have a high blood alcohol content and that region of your brain isn't working properly. Proprioception, equilibrium, and balance. The moment that you ask somebody to close their eyes and carry out a task, so let's say finger to nose, fine-tuning skeletal muscle contraction, the moment that you say close your eyes and bring your finger to your nose, you're also evaluating that person's ability to carry out proprioception. And that's another cerebellar activity. Walking heel to toe is equilibrium and balance. That's another cerebellar activity. So people who have true cerebellar damage often present motor-wise, right, with the way that they're moving their body, like they're drunk even though they haven't had anything to drink. The field sobriety test didn't originate first. That was a clinical test to evaluate cerebellar function, which then turned into a test that was useful in law enforcement. So I want you to see what a real cerebellar cue would look like. And let's watch this video real quick. It's a very quick video of this individual who has cerebellar dysregulation. And let's just look at this guy.
And postural tremor is intention action tremor seen with cerebellar
look
disease.
At this side of the body.
In this
Remember,
patient who has had a traumatic hemorrhage involving one side of his cerebellar white matter pathways, he has a normal function on the right side, but the left side shows significant dyssynergia.
More importantly, watch what's going on.
This type of tremor contrasts, however, with the postural tremor, in that both the trajectory and the endpoint
finger to nose test, quintessential cerebellar test,
is in fact right at the endpoint when he tries to touch his nose or to bring his finger totally out.
What he's having trouble doing is fine-tuning skeletal muscle contraction, which is a true cerebellar sign, right? And in this case, it was cerebellar white matter pathways, but you know that something is going on with the cerebellum. That is a specific test for cerebellar function. So going back to our PowerPoint, so the finger to nose test is really an important test for cerebellar function. So now we've gotten done talking about the cerebellum. What we're going to shift our discussion to is the cerebellum. And when you look at the cerebellum, a couple of structures I want you to know within the cerebellum. First off is the white matter pathways in the cerebellum are called the arbor vitae. Arbor means tree and vitae means life, so these white matter pathways are called the tree of life or the arbor vitae. They are specific structures within the cerebellum that allow for the transmission of information. The gray matter structures of the cerebellum, meaning this gray matter area at the outside or the external regions of the cerebellum toward the periphery, is called the cerebellar cortex. Don't confuse the cerebellar cortex with the cerebral cortex. So these are two specific structures within the cerebellum you need to know. Now, fine-tuning skeletal muscle contraction. So we're going to continue moving up, and what we are going to focus on is the diencephalon. Now, the diencephalon is this entire brain region, right? It's a major region of the brain that has three substructures. The substructure indicated by orange is the thalamus, by pink here is the hypothalamus, and by red here is the epithalamus. We're going to start our discussion of the diencephalon. So if I circled this region and asked you to identify the major region of the brain, that'd be the diencephalon. If I pointed at this and asked you to identify the structure, the first structure within the diencephalon that makes up the diencephalon that we're going to talk about is the thalamus. And what the thalamus is a relay center for both sensory and motor information. It functions as a relay center. It's like a 1920s operator. So you call the operator and say, hey, give me Mr. Brown. And they take that and they rewire it and route you to that particular phone extension, right? That's what the thalamus does. It's a routing center for information that takes that information or routes that information to the appropriate brain structure where that information is supposed to go. So it functions as a relay center for sensory and motor information. Now, when you look at the thalamus, it's made up of a series of 9 nuclei, none of which you have to know. But notice that the thalamus has outputs to every single region of the brain. It's connected to every region of the brain. And what essentially happens is, let's say you have information coming from your inner ear, right? What we think of as being sound. Well, that's an electrical signal that travels along a neuron, and ultimately that information will end up at the thalamus, and it will synapse with neurons in a nuclei that makes up the thalamus, in this case the medial geniculate nucleus, and then that information will be routed to the auditory cortex. Or information coming from the eye will go to the lateral geniculate nucleus and be routed to the visual cortex. Or information coming from the sensory receptors of the skin will go to the somatosensory cortex. It is a relay center for sensory information. So when you see something, an action potential is triggered, travels along your optic nerve, and the neurons that make up that optic nerve, right, synapse with neurons in the thalamus, which ultimately transmit that information to, for example, the visual cortex, where that information is processed. It is a relay center for information. Something I want you to think about is, let's say that we did that wiring a little bit weird. And let's say that most of that wiring, right, from the ear is supposed to go to this medial geniculate nucleus, but just a few stray fibers go to the lateral geniculate nucleus. Well, because all of your sensory perceptions are a construct of the mind, if you have electrical signals that are being triggered and then information going to the visual cortex, you could potentially do something like see sound. And that's, the thalamus has been linked to a really interesting condition called synesthesia. And we don't know, those particular pathways are complex, and there's debate about the exact mechanisms by which they happen. But it's just interesting to think about that wiring and to think that we can have these conjoined senses, like you can taste touch, or you can smell color, or whatever it is, whatever conjoined sense. And a lot of people are synesthetes, and if you want to watch a little bit more about that, there's this great video by David Eagleman, a neuroscientist that I happen to really enjoy, that I've put up on YouTube. The hypothalamus. Now, the hypothalamus is a constellation of a ton of different nuclei. We're not going to focus on each one. We're just going to talk about the general functional roles of the hypothalamus and tie it to some things you might care about and talk about the mind-body connection. So the hypothalamus is a master regulator of autonomic function. A lot of your autonomic function has overarching control from the hypothalamus. So the hypothalamus communicates quite closely with brainstem structures to maintain things like heart rate, blood pressure, respiratory rate, etc. Really important master regulator of autonomic function. The hypothalamus is also a master regulator of endocrine function. So when the nervous system and the endocrine system interact with one another, we call it a neuroendocrine interaction. Now, the nervous system and the endocrine system interact quite frequently. They are two systems of homeostasis. The point at which you see the most important interactions are between the hypothalamus, which is a brain structure, and the pituitary gland, which is an endocrine structure. And the hypothalamus essentially regulates the activity of the pituitary gland, and that's really, really important. In that respect, it essentially modulates many of the functions of the endocrine system. So we have neuroendocrine regulation, thermoregulation, the regulation of temperature takes place in the hypothalamus, hunger. The hypothalamus has neurons that dip into the blood. It's the only brain structure that can come into contact with blood that monitor levels of like glucose and amino acids. Thirst, the hypothalamus can monitor blood concentrations to determine whether they get too high. If blood concentrations get too high, the endocrine and behavioral responses associated with thirst will occur. Sexual arousal. It's part of what's called the limbic system. The limbic system is a grouping of structures that coordinates the behavioral state of emotion, what we think of as being emotion. Are you angry, happy, sad? So the hypothalamus is really, really important, super important. Now, when we think about the mind-body connection, I want to emphasize how important this regulation of the endocrine system is, right? The hypothalamus is part of the limbic system, meaning it's connected to other brain structures that regulate the behavioral state of emotion. So let's say that I'm anxious and I'm stressed because I have something like an exam coming up in an anatomy and physiology class, and I'm like, I hate that guy. So that information is going to come in, it's going to be processed by different areas in your brain, and ultimately a message is going to be sent to the hypothalamus that says, hey, we're stressed out. The hypothalamus is then going to trigger a signal, what's called a releasing hormone, that travels to the pituitary gland, and it's going to say, hey, pituitary gland, release this hormone called ACTH. ACTH is then released by the pituitary gland. Hormones are chemical messengers released directly into the blood, and then they travel to distant target cells, in this case the adrenal glands, and specifically the adrenal cortex, where they tell the adrenal cortex to release a hormone called cortisol. Cortisol does two things that I want you to be familiar with. One, it increases blood glucose, and two, it suppresses your immune system. So it jacks up your blood sugar and suppresses your immune system. So now, a series of thoughts taking place in your mind where you think it's kind of restricted to that area in your cranium is no longer restricted to the cranium. That series of thoughts has now triggered a set of events that have changed the biochemistry of your body. So the mind and the body aren't just linked philosophically or, metaphysically. They are linked in a very real, right, structural and biochemical way. When you get stressed out, you release the stress hormone cortisol. Cortisol jacks up your blood glucose, lowers your immune response, and it makes you more prone to diseases like diabetes, to common coughs and cold, to opportunistic infections. It can, promote things like obesity. So there is a very real link between what's happening in your mind and the biochemistry of your body. And one of those things that's really important to realize is sometimes you just got to go and bring it down just a little bit. There's a great book written on this called Why Zebras Don't Get Ulcers. Also a great TED Talk about that book. So that's one I want you to understand about the hypothalamus. So we have the hypothalamus. Then we have the epithalamus. Now, the epithalamus consists of a bunch of different structures. We're not going to talk about some of the nuclei and some of the tracts, but we are going to draw our focal point onto the pineal gland. And the pineal gland is largely responsible, or to a degree responsible, for regulating what are called circadian rhythms, our sleep-wake cycles. So when it gets dark out, the pineal gland starts to produce a chemical messenger, we'll call it a hormone, called melatonin. And melatonin peaks when we're in deep sleep, and that melatonin promotes a relaxed brain, meaning it promotes sleep by promoting a relaxed brain. It shuts down different regions of the brain, or it kind of calms them down a little bit prior to sleep. Now, as we get closer to the morning and sunlight comes up, melatonin dips, and what spikes? Well, that's our stress hormone cortisol. Then you wake up and go, damn it, another day at ACC, damn it, another day of the coronavirus. Your stress hormone spikes, and then it goes down throughout the day, melatonin, and you get this cycling. So the pineal gland is really important in regulating circadian rhythms because it produces a chemical signaling molecule called melatonin. Now we're on to the cerebrum. Now, I know that these lectures are long and you can skip through them and you can drag through. Remember that I've been given 1 week to convert 5 classes to an online format, so I'm trying to do this as quickly as I possibly can and as effectively as I can. I would make a series of little videos for you, but I just don't have the time to do it. Now, when we look at the cerebrum, which is what I consider our higher order brain structures, is broken into 2 hemispheres. And there's a fissure called the longitudinal fissure that essentially forms the demarcation or the delineation, the border between one hemisphere and the next. And that fissure right there, again, is called the longitudinal fissure. So if I was to say, identify the structure indicated by the line, I'd be looking for the longitudinal fissure. Now, when you look at the cerebrum, if we were to take a small frontal section of the cerebrum and look at it, we have our cerebral white matter, which is made up predominantly of myelinated axons transmitting information from point A to point B. And then we have our cerebral gray matter called the cerebral cortex. Now, remember, gray matter is made up predominantly of dendrites and neuron cell bodies. So dendrites receive information and neuron cell bodies process that information. Humans have proportionate to their body size, have one of the thickest cerebral cortices in the animal kingdom. And does that have functional significance? It does. We have a lot of gray matter in our brain, which allows us to process a lot of information and results in a lot of interesting emergent characteristics with respect to our cognitive capabilities, our abilities to carry out complex tasks. So, and to imagine and to dream and to abstract, etc., etc. And I won't go too on about that. So when we do the sheep brain dissection, you're going to notice the cerebral cortex isn't quite as thick as that as you see in the human brain, because sheep are not capable of the same cognitive tasks. Gray matter is important. The amount or volume of gray matter you have is important. There's a correlation between gray matter volume and cognitive capabilities. Now, when you look at this, the brain is a very large structure in a very small space, the cranial cavity. So in order to fit all that gray matter into this restricted little space of the cranial cavity, we have to fold the brain up. So think of your brain kind of like a newspaper, and think of all the information it can store, but it doesn't take up a ton of room because it's folded. And these folds in the brain, the elevations of these folds singularly are referred to as gyrus, plurally are referred to as gyri. And the depressions of these folds are referred to singularly as being sulcus or a sulcus, or plurally as being sulci. So when we talk about specific gyri and sulci, what we're talking about are the elevations or the depressions in the folds of the brain. When you think about animals that have lots of gyri and sulci, it means their brain is highly folded, like mammals, and that often translates into interesting cognitive characteristics that we kind of think of as being intelligence. So lots of folds in the brain are a very interesting anatomical correlate in the brain with respect to cognitive function. Now, there are some prominent sulci. This sulci that separates essentially the anterior portions of the brain from the posterior portions of the brain, this prominent sulcus right here is called the central sulcus, and I'll point that out again, but that's essentially a delineation between anterior and posterior regions of the brain. And just like the spinal cord, where the anterior and posterior regions serve different functional roles, the anterior and posterior regions of your cerebrum serve different functional roles. We also have our left and right cerebral hemispheres. So if we were to review this, if I was to say, identify the structure indicated by the line, that would be the longitudinal fissure. Identify the structure indicated by the pointer, that would be a gyrus, a sulci. Identify the gray matter structure indicated by the pointer, that would be the cerebral cortex, and I would probably outline the whole thing. Identify the structure indicated by the pointer, that would be the cerebral white matter. And if I said identify the hemisphere of the brain indicated by the pointer, this would be the left and this would be the right hemisphere of the brain. Now, when we're mapping out the brain, right, we've got that basic functional mapping. And we have our left hemisphere of the brain and the right hemisphere of the brain, and oftentimes the functional areas of the brain are symmetrical, meaning that there's a motor cortex in the left and a motor cortex in the right hemisphere of the brain. But there are different functional areas of the brain that are unique either to the right or to the left hemisphere, and we refer to the fact that there are unique functional areas in the brain either in the left or the right hemisphere as being what's called hemispheric lateralization. So for example, many of our language and mathematics centers are in the left hemisphere of our brain, while many of our spatial visual reasoning centers are in the right hemisphere of our brain. As a consequence of that, the two hemispheres of the brain need to communicate. And the way that they communicate is along a white matter pathway called the commissural tract. And this white matter pathway that allows one hemisphere of the brain to communicate with the other hemisphere of the brain is called the corpus callosum. So the structure I'm indicating right here, this big, thick white matter pathway that allows one hemisphere of the brain to communicate with the other hemisphere of the brain is called the corpus callosum, right? So it's called the corpus callosum. And it's really, really important. Communication between the right and the left hemisphere of the brain is extraordinarily important. Now, when we think about communication between the right and left hemisphere of the brain, remember the left hemisphere of the brain has areas unique to language and mathematics, or certain types of arithmetic or mathematical processing. And the right hemisphere of the brain has regions unique to certain types of spatial visual reasoning. We sometimes think about people who are left-brained, which is a total misstatement. I mean, that whole, like, you can only use 10% of your brain, that's completely false. That was from a psychological study, I think, from the, 1960s. It just got blown up by the media, and now people think of it as being common knowledge. We use way more than 10% of our brain. We just use it in different ways at different times. And the ability to use 100% of your brain because some Chinese gangster kicked your stomach because when you had this weird party drug and it gave you superhuman powers, you can't manipulate the fundamental fabric of space and time. I find that premise hard to swallow, if anybody knows what I'm talking about. So, what you're looking at here, this little bridge between the left and the right hemispheres of the cerebrum, that is the corpus callosum, and that's where information is being exchanged, and that exchange of information between the two hemispheres is really, really important. So if you want to, you can go back and you can watch the video I've embedded in this PowerPoint, which essentially walks you through the set of experiments that led to the discovery that information is exchanged between one hemisphere of the corpus callosum and the other. And in this video, what they end up doing is severing somebody's corpus callosum to prevent seizures, and then you get to see all of those interesting kind of functional consequences of severing the corpus callosum. Now, the cerebrum is broken into a series of what are called cerebral lobes. On an exam, if the answer is a cerebral lobe, like the frontal lobe, the parietal lobe, the occipital lobe, the temporal lobe, and embedded, because the brain is so folded up between the frontal, parietal, and occipital lobes, if you were to peel back this brain area, you would see another lobe of the brain called the insula. If I'm asking you to give me a cerebral lobe, right, which is just a big chunk of cerebrum, I will ask you identify the cerebral lobe indicated by the pointer, and the cerebral lobes are named after the bones that rest on top of them. So that should give you a pretty good idea of what you're actually looking at with these cerebral lobes. You'll get maybe one or two of those questions where I just say identify the cerebral lobe indicated by the pointer. More important to me than cerebral lobes are mapping out functional areas of the brain. And there are going to be a few functional areas of the brain that we go over in this discussion. What you have on your handout to follow along with what we're talking about right now is an image like this. So remember that when you're taking this on the computer, you are going to be able to see that color, and that this particular image is going to come back to haunt you on the exam. So sensory areas of the brain have direct sensory inputs coming from the peripheral nervous system. So for example, the auditory cortex, information coming from the inner ear is routed to the thalamus directly to the auditory cortex, and that's what constructs the conscious perception of sound. The visual cortex has information coming along the optic nerve from the eye to the thalamus to the visual cortex, direct sensory inputs. The somatosensory cortex, receptors like Meissner's corpuscles, Pacinian corpuscles, route that information to tracts within the central nervous system that go to the thalamus, like the spinothalamic tract, which is ultimately then routed to this area of the brain called the somatosensory cortex. And these are responsible for constructing the conscious experience of the sensory stimuli that we come into contact with. Now, we're not going to get that much into association areas because, again, we're running on a timeline. Motor areas of the brain have direct motor outputs. So we talked about the premotor cortex as planning skeletal muscle contraction and the primary motor cortex initiating skeletal muscle contraction. The primary motor cortex would be considered a motor region of the brain, while the premotor cortex would actually be considered a different type of area that we're going to talk about in just a bit. So primary motor cortex, direct motor output. So think about that picture you had to draw where you're drawing the upper motor neuron synapsing with the lower motor neuron, which ultimately goes out to the neuromuscular junction and controls muscle. We have direct motor outputs. If you activate the primary motor cortex, assuming a healthy system, you're going to get contraction of a muscle. The Broca's area is involved in the muscular contractions associated with speech. It allows you to physically actually speak. Association areas. Association areas don't get a lot of direct input or have direct motor outputs. They are where information is processed and made sense of. And they're what you consider to kind of be thought areas, meaning what happens in association areas, even though it can influence motor and sensory areas, is largely restricted to the brain. It's an internal experience. So the Wernicke's area, which I would never ask you to identify, anatomically, is responsible for understanding language, right? We'll think of it very generally because we're in a big hurry today, but it's responsible for understanding language. Premotor cortex isn't responsible for initiating skeletal muscle contraction. It plans skeletal muscle contraction. And the prefrontal cortex right here is responsible for what are called executive functions, what we think of as being higher order thought. So we'll talk about that or elaborate on that. So we have the prefrontal cortex, premotor cortex, primary motor cortex, primary somatosensory cortex, primary visual cortex, primary auditory cortex, Wernicke's area, and Broca's area. So let's just go through those one by one. Prefrontal cortex. Well, we learned a lot about the prefrontal cortex from Phineas Gage, and what you're looking at here is actually the skull of Phineas Gage because he was so important in neuroscience and to our breakthroughs that have happened in neuroscience. When you think about the prefrontal cortex, one of the things that the prefrontal cortex is responsible for is executive function, which is goal-directed behaviors. Things like decision-making, initiating control over deliberate actions, targeting your attention when you're studying, problem-solving. For example, if your flashlight runs out of battery or is not working and you go, is it out of batteries or is the bulb out? Planning and initiating activities. Social behavior and reasoning. So the reason you don't show up to my class wearing your boxers, smoking a cigar with your feet kicked up on the table is because you've learned through social conditioning that that particular behavior is not acceptable or socially acceptable. As a consequence of that, even though you may have the urge to do that, you have this big, thick prefrontal cortex that essentially runs that information through a simulation and goes, no, the outcome of that isn't going to be very good. And that's collectively what we think of as being executive functions, just the ability to control your behavior, problem solve, think about things. Another interesting characteristic of the prefrontal cortex I want you to know is that it's a simulation device, right? Meaning that we can simulate events within our prefrontal cortex that haven't happened, and we can predict the outcomes of things that we haven't done, which is a pretty interesting capability. So for example, the reason that we don't have garbage flavored ice cream is we haven't actually made it, or maybe somebody has, right? But we know that garbage probably wouldn't taste very good, so why put the time and effort into making garbage-flavored ice cream if we could just predict using our brain that it's not going to taste very good? It's really, really important. This executive function, right, is really important, and it's a lot of what we think of as being our personality. So it's a really important functional area of the brain. Think executive function when you think prefrontal cortex. Now, the premotor cortex, as we discussed when we went over the muscular section, is responsible for planning skeletal muscle contractions. The primary motor cortex, so neurons in the premotor cortex get routed, communicate with the basal nuclei, which ultimately feeds information to the primary motor cortex. The primary motor cortex is responsible for initiating skeletal muscle contractions. So premotor communicates with primary motor. Any region of the brain that has direct inputs or outputs to the body, in this case we're talking about motor outputs, will always have a little map of the body in the brain, because there needs to be a way to organize those neurons to make sure they're getting to the regions that control the specific areas of the body that they're supposed to. This little map of the body that's disproportionately represented with different structures in our body, this little map of the body in the brain is called the motor homunculus. The little map is called the homunculus. And what the motor homunculus does, notice that the areas of the body are drawn disproportionately. So the hand takes up more space in the brain, meaning it takes up more cortical gray matter to control the hand than it does the entire rest of the body. Why is so much of your brain dedicated to moving the hand, right, and so little of it dedicated to moving, for example, the trunks and your trunk, your torso, and your legs? Well, it's because the hand has a lot of fine-tuned movements. There's a lot of neurons associated with it, so there needs to be a lot of input going to that hand. There are a lot of neurons that actually control the muscles that move the hand, and that's reflected in this motor homunculus. So if you ask somebody to start wiggling their fingers on an MRI, where you're going to get the light up on the MRI or the fMRI is in this region of the motor homunculus right here. So what that also does is it prevents overlapping pathways. So you can have descending pathways that aren't going to overlap because the brain is organized in such a way that those neurons are not, it prevents the overlapping pathways or descending pathways. So that's our motor homunculus. Premotor cortex plans muscle contraction. Primary motor cortex initiates muscle contraction. The primary somatosensory cortex, this is where somatosensory information is initially routed and processed. So we determine, is that pain and where is it coming from? Is that touch? Where is it coming from? Is that tickle? Where is it coming from? So information coming from, for example, the sensory receptors in your integument, like free nerve endings, Pacinian corpuscles, Meissner's corpuscles, all of that information is routed to the primary somatosensory cortex, where it's initially constructed into conscious experience. You go, oh, somebody's touching my arm, well, or forearm. You don't go, sensation doesn't occur on your arm. This is just the beginning of a pathway. All sensory perception is an artifact of the brain. It's a construct of the mind. So you don't get sensory perception here. You're getting sensory perception here. And where that information is constructed, that somatic sensory information that tells you where that information is coming from and what type of information it is within the primary somatosensory cortex. So just like that motor homunculus, there are sensory homunculuses, or little maps in the brain that prevent overlapping pathway and let your brain know, hey, this is where this particular sensation is coming from in the body. Now, one of the things I want to highlight, and this is just a brief tangent that I'm going to take on mental illness, is this was a PET scan, which is essentially where you look at the distribution of this radioactive glucose in the brain and you can kind of make the inference that that's a spike in brain activity. But this is a PET scan of somebody who's schizophrenic. And schizophrenic disorders tend to manifest as hallucinations. Hallucinations can be visual, they can be auditory, they can be somatosensory. So the individual in this study felt like there were things crawling on their skin all the time, right? And even though there was no stimuli actually on their skin, their brain activity reflected that their somatosensory areas were kind of spiked with activity. What I'm really saying is that you can't project your mental state onto somebody else, because you never know what's going on with somebody else's brain. And mental illness, right, is a brain that's not functioning properly, whether it's not producing an adequate amount of a neurotransmitter, or there are regions that aren't connected properly, or there are nutritional factors that are interfering with the functionality of the brain, or there's just anatomical factors where that brain is set up weird. You can't think about yourself when you're thinking about somebody who has an overactive brain, like somebody suffering from schizophrenia, because their plane of consciousness is different than your own. And it's something that if you're going to be a clinician working with people who have mental health issues, you really have to understand that everybody kind of exists within their own plane of consciousness. Now, the visual cortex. Information coming from the retina of the eye is routed through the thalamus to essentially what takes up the entire occipital lobe, which is the primary visual cortex. And the visual cortex is responsible for constructing the conscious sensation of vision. It takes up a huge percentage of the brain, and we could go on all day talking about the visual cortex. I won't bore you with this study at the moment because we've been at it for a while today, but it's a really important brain structure. The primary auditory cortex, as you can probably put together, is responsible for constructing the conscious perception of sound. Sound doesn't happen in the ear, it happens in the mind. Now, ancillary functional areas that I want you to know. So other functional areas of the brain I want you to know. And again, we're going through this in one fell swoop. I know it's a long lecture. I should break these into shorter lectures. I will in the future when I have a bit more time. But what we need to know now. The Wernicke's area, which is a functional association area of the brain, plays an important role in allowing us to understand language. So the Wernicke's area plays a really important role in allowing for the understanding of language. And the understanding of language, it could be when you're reading a book, it could be hearing language, it could be Braille, right? It could be sign language, but language, complex language, is largely processed within the Wernicke's area. So that information comes into the Wernicke's area, and the Wernicke's area, which allows you to understand language, has outputs to what's called the Broca's area. Now, even though this is a horrible simplification, right, we're going to break it down or distill it to its essence. The Broca's area is the area that plays a moderate role in understanding, but plays a much more important role with respect to being able to actually initiate the skeletal muscle contractions associated with speech. So the Broca's area allows you to physically speak. So whereas the Wernicke's area allows you to understand language, the Broca's area allows you to speak, right? It allows for the muscle contractions necessary to formulate speech. Now notice that we're looking at the left hemisphere of the brain. The Wernicke's area and the Broca's area tends to be located exclusively in the left hemisphere of the brain, right? Now, there are a lot of interesting studies with kids who've had hemispherectomies, and that's way beyond my pay grade. So we'll just talk about it in general terms, how it applies to many people. Now, there's a condition called aphasia, A-P-H-A-S-I-A, and aphasia is having trouble or difficulty speaking. Now, two types of aphasia, or two general categories of aphasia, is we have what are called Wernicke's aphasias, or we can have what are called Broca's aphasias. In Wernicke's aphasias, the Wernicke's area is damaged, and how that typically manifests is with trouble understanding speech, but being able to speak with a relatively normal cadence and tone. So let's say you had somebody with a hemorrhage or a stroke, an aneurysm or a stroke that destroyed significant portions of their Wernicke's area. You were to ask them a question. What's your favorite color? And they go, chicken, horse, cowboy, monkey. They're still able to speak, but what they're throwing together are what are called word salads. And that kind of word salad presentation is a hallmark presentation of Wernicke's aphasia. They can, still speak, but they're having trouble understanding what's coming in. The Broca's area, on the other hand, even though it does play an important role with respect to language comprehension, it does, and it's a complex area. I'm just speaking in generalities right now. But one of the functional roles of the Broca's area is it's a motor area that allows you to physically speak. So if you were to completely destroy the Broca's area, you might understand what's coming in, and you might be able to write it down, right? And there's caveats to that we're not going to get into, but you're not going to be able to physically speak. And when you can't physically speak, or when your speech patterns are really interrupted, like you're getting rid of intermediate words like the, went, park, like that, instead of went to the park, that's a hallmark sign of Broca's aphasia. So let's see if we can figure out what this dude was going on with this dude.
Their foot, boom.
What's this?
The big end right there. End of the football.
End of the football?
Yes. Is it it influences the football right here. The cause that it comes in, see? Yeah. The end of it, see? Yeah. Oh, we have a lot of things, see?
I don't understand.
So what do you think? Broca's or Wernicke's aphasia? And take a stab at it. You're more than welcome to email and ask me, but I bet you if you're intuitive, you can probably put that together. The final set of brain structures we're going to talk about are associated with what are called the limbic system. We call them limbic system structures. And the limbic system is what we think of as being our emotional brain. Now, the limbic system isn't just one brain structure, it's a series of intercommunicating brain structures. I got a fuzzy graph or a fuzzy image intentionally because I don't want you memorizing every structure associated with the limbic system. But when you talk about the limbic system, you're talking about a grouping of structures that communicate, that regulate emotion in the brain. You want to get somebody to pay attention to what you're saying, activate their emotional response, because then their limbic system is cued. And once you got the limbic system, you've got somebody, right? So the limbic system is the emotional brain. Now, there are a couple of really important structures within the limbic system, but we're going to talk about two. We're going to talk about the hippocampus and the amygdala. Now, the hippocampus is responsible for storing short-term memory and routing long-term memory, meaning deciding on what's going to be stored as long-term memory. It's called long-term memory consolidation. So the hippocampus can store, for example, when I say short-term memory, when you're just remembering a phone number for a few minutes that you're just going to forget in a couple of minutes, or you set your keys down, that's short-term memory. When I say it's responsible for consolidating long-term memory, I say, what I'm really saying is, let's say you're exposed to something like 15 times and you don't remember it the first 14 times, like a bone marking. But on that 15th time, you see it, your hippocampus recognizes it again and goes, oh God, we've been exposed to this over and over again. And it routes that information to areas in the temporal lobe that are responsible for long-term memory consolidation. Right? Meaning storing that memory, not just for the short term, but for the long term. So that's why I constantly say studying 2 hours a day is better than studying 10 hours in one day, because you're exposed to that information more and more. And what your hippocampus is going to do is ultimately route that information once it's exposed to it enough times, or once there's enough repetition, to your long-term memory storage areas. And that's a good thing. The hippocampus is a group of subcortical nuclei that kind of looks like a little seahorse. And that's that guy right there. I really like the hippocampus, actually. I think it's very interesting. At the tip of the hippocampus, we have another structure called the amygdala. Now, the amygdala is responsible for applying an emotional quality to stimuli. I'll say that again. The amygdala is responsible for applying an emotional quality to stimuli. So let's do an amygdala exercise right now. What am I? What am I? What am I? As you look at that, that's just reflected light coming through to you on a YouTube video. But the first one, you could have gone, oh, he looks really angry. The second one, you could have gone, oh, he looks happy. The third one, you could have gone, he looks contemplative or constipated. I don't know, right? One of those two things. So it applies an emotional quality to stimuli. That's what the amygdala does. It applies a basic emotional quality to stimuli. So when you see something like a smiling person walking at you, it doesn't trigger a negative emotion in you, and you go, oh, great, I'm going to see that person, as opposed to if you see somebody with a knife walking at you. Even though they're similar stimuli, you're able to apply an emotional quality to that. The amygdala is tightly linked to inappropriate emotional responses, such as anxiety or fear. So people with post-traumatic stress disorder, for example, when they're exposed to a benign stimuli, let's say like a loud bang, right? They get an amygdala response that's much greater than somebody who doesn't have post-traumatic stress disorder, because that's a trained response. So I had a student in one of my classes who served in Afghanistan, and she drove Humvees, and she wouldn't drive anywhere near white lines on the freeway. And I said, that's interesting. How come? Well, because that's where IUDs used to be placed. And I said, oh, well, that seems like a perfectly reasonable fear then. Now, you wouldn't fear that just as an everyday thing, but that white line triggers an emotional response, and that's the amygdala saying, danger, danger. It applies emotional qualities to stimuli. It allows you to determine whether somebody's happy, sad, whether something's scary or benign. So it's a really important part of your limbic system. It's essentially the root of your limbic system that activates your limbic system. Now, the basal nuclei, we refer to these as being subcortical gray matter structures. They're just a series of nuclei located deep to the cerebral cortex. And the basal nuclei play a really important functional role in a lot of different things, but we're going to think about movement. The premotor cortex and the primary motor cortex have intermediate communication in the basal nuclei, which can kind of smooth out and help coordinate movement and control movement. It also plays a role over overarching or regulating behaviors. It's kind of like a filter in your brain. So you may think about something in your prefrontal cortex like coming and punching me in the face, and you may even imagine actually doing it. But when that information gets routed to the basal nuclei, it can damp down some of those particular, that neuronal activity, and it can correct your action to be more socially appropriate. So people with things like Tourette syndrome, obsessive compulsive disorders, oftentimes have problems with the basal nuclei. So it's kind of like a filter for the brain, and it's also really important with respect to regulating movement. And that's as deep as we're going to get into that. Have a good day, and thank you for listening to this lecture. Bye-bye.